I am pleased to present my research on Baronia brevicornis, and thank you very much, Fabienne and Martin, for your very kind invitation. Leonida Vasquez, a Mexican researcher, made significant contributions to the understanding of Baronia brevicornis. Since her descriptions, studies exploring various aspects of its natural history, ecology, and genetics have proliferated, and here I show you some examples about, about these topics. But one intriguing aspect of this butterfly is that in some populations there are 3 female morphs and 4 male morphs. They differ in color. Additionally, Baronia brevicornis is considered the oldest butterfly of the family Papilionidae. However, despite these interesting topics, it is classified as endangered. According to Luc Legal and his colleagues, this relic species is the most enigmatic butterfly in the world. In this video, I will show you some basic knowledge about Baronia. The butterfly inhabits tropical dry forest. The larvae feed on Bacteria campechiana and create 2 large shelters by binding leaves with silk and thereby protecting themselves from predators. After 1 month of development, the larvae pupate and remain underground for 1 to 2 years. Upon emerging, the insect becomes a sexually mature adult, and after maturation, the adults climb to the top of Acacia plants. Once males are fully developed, they compete to access to the tips of the Acacia plants because females visit these plants to mate and oviposit. Males court the females for a few seconds, I'm showing here. And if the female is receptive, they lift the abdomen to facilitate mating. Adults have a lifespan of about 2 weeks, so most of the time Baronia remains buried. The topics that we are studying in Baronia are regarding evolutionary ecology. We are studying the invertebrates' immune memory and sexual selection. And regarding conservation, we are monitoring the Baronia populations since 2015. In this talk, I will focus my results in the invertebrates' immune memory and the monitoring program. The immune memory in invertebrates refers to the protection and improvement of survival after a subsequent encounter with the same parasite or pathogen species or strain. So immune memory should be very specific. This phenomenon is a shift in paradigm, as it was previously proposed that only vertebrates, not invertebrates, possess immune memory. This field of research is relatively new, since the seminal work of Joachim Kurtz and Caroline France. Very few papers have been published. In a couple of reviews by my laboratory, we found only between 120 and 140 papers in the last 20 years. Therefore, we inquire if dual homologous challenge, memory, provide better protection to larvae than dual heterologous challenge. Baronia brevicornis was exposed to Micrococcus lysodecticus, and Tom Little and his collaborators proposed a method to test immune memory, which include a comparison between homologous and heterologous groups. In our study, larvae were challenged twice. First, with a sublethal dose of Micrococcus lysodecticus, which was a dose of 0.5 milligrams per milliliter, followed by a second injection of the same bacteria after 7 days, but the second injection consists of 2 milligrams per milliliter. And the control group underwent a lethal challenge with micrococcus. So survival and the immune response were recorded after the second challenge. To assess the immune response, we recorded the lytic activity. This animation demonstrates how antimicrobial peptides in green break down pathogenic cells. And the next video depicts insects' hemocytes surrounding a nematode, attacking it and subsequently producing melanine, which is very toxic to pathogens and parasites. Simultaneously, during this process, insects generate reactive oxygen species or pro-oxidants to eliminate the invaders. So, very good parameters of the immune response are the lytic activity, phenyloxidase, and pro-oxidants. Considering the percentage of organisms that survived 24 hours after the second challenge, the experimental group, the memory group, survived better, 81%, compared to the control group without memory, which was 42%. Therefore, the immune protection enhanced survival akin to the effects of vaccines in vertebrates. Although we didn't observe significant differences between immune memory in control groups in terms of lytic activity and pro-oxidants, the experimental group exhibited higher levels of phenyloxidase compared to the control group. This suggests that that immune protection enhances the melanization response. Certainly, further research is needed to test the performance of immune response against different parasites and pathogens, because my laboratory has shown that the role of parasites is very important for the immune memory outcome. And to elucidate, it's important to elucidate the mechanisms that control immune memory. Given the phylogenetic position of Baronia brevicornis, it's intriguing to explore the evolution of immune memory in this group, taking into account the underlying mechanisms. The new memory evolution within these papillonids raise fascinating questions. For example, as if they share the same molecular mechanisms. All of these groups present immune memory, or just some of them. These kind of questions will provide some information about the evolution of the immune memory, not only in vertebrates but also in invertebrates. So I'm eager to address these questions. Fundamental questions within evolutionary ecology can be explored in Baronia. However, it's essential to consider the challenge they may face in nature. So, we have been monitoring a population since 2015, and throughout this period, we documented their host plants, population sizes, and morphs. And we also are investigating the influence of precipitation and temperature on the capture rates. This is on the probability to observe butterflies in the field. It was previously thought that, or hypothesized, that the distribution of Baronia might closely align with the distribution of its larval host plants. However, our observations have shown Baronia in areas where food sources for both larvae and adults are available. And interestingly, we didn't observe organisms in locations solely occupied by acacias. In this video, I will show you adults feeding on Pithecolobium dulce and adults feeding on Bursera copalifera. But we also found another three plants in which adults were feeding on. We also corroborated the presence of pollen on the buccal apparatus of these butterflies. So, in the forthcoming years, we aim to investigate the influence of various host plants on the distribution of Baronia. Every year, for or every 2 years, we collect butterflies, mark them, and then release them to record their numbers day by day during the adult season. And every year, we have observed fewer females than males at the mating sites, although the sexes emerge in a similar proportions. In blue, I show you the percentage of males compared to the percentage of females in green. So we don't know if whether females die before males or if they simply mate and then move to another location. In our laboratory, adults emerge after 1 and 2 years of pupation. However, the percentage of individuals that emerge decrease in the second year compared to the first year. In the first year, we observed a little bit more than 75% of adults emerging from the ground. And for the second year, we only observed a little bit more than 25%. This could be due to the cost associated with prolonged diapause. So we will explore these topics in the future. Now I show the presence of the frequency of morphs for males and females across different capture years. And we observe that the frequency of morphs can vary depending on the year of capture. For males, in 2021, we observed a little bit more the brown morph with orange spots on their wings. And for females, on 2021, we observed a little bit more females of the orange morph. So this indicates that the morphs are not stable over time. To determine the effect of temperature, precipitation, and year on the number of captures for each season or the probability to observe adults flying, we used a generalized additive model. This model, capturing both linear and non-linear effects, allow us to estimate the contribution of each predictor variable, the x-axis, to the total number of captures, the y-axis. We were collecting butterflies in different years, and this study was performed by Yesenia Alvarado, my student, and the statistical analyses were carried out by my wife, Gloria Ruiz. We did not find any effect of temperature or the maximum temperature, which ranked between 26 to 38 centigrade, because um, above of the dashed line, which is shown around 0, results are significant. And in this case, the dashed line and the probability of capture organisms are aligned. However, we observed that in some years there were no adults flying, and hence we didn't observe Baronia, so they remained underground. And this was particularly observed in 2018. You can see how the number of butterflies were below the dashed line. And the opposite was found for 2021 because we observed many butterflies above of the dashed line. It's important to mention that for 2018 it was a particular dry year, so we hypothesize that the probability of emergence of butterflies and hence to capture them in the field is related to precipitation. And accordingly, we found that the effect of accumulated precipitation on captures is positive and significant between 90 and 170 millimeters, while lower values such as 50 millimeters or higher values such as 300 millimeters leads to a decrease in the number of captures in the field. So it seems that there is an optimum of capture rates in the field due to precipitation, and this optimum may be related with 120 and 140 millimeters. So in the next year, we would like to investigate the importance of the abiotic factors on the number of butterflies. Because this year is uncertain, because it's too dry, and butterflies should be flying at this time, but my students have not observing any butterfly yet. It might be this year, it might be just like in 2018, in which we didn't observe any butterfly in the field. I would like to thank many people involved in these studies. I began my research journey alongside Luke and Marcela. Subsequently, Yvette Galicia joined us, but sadly she passed away during the course of our research. Jorge and Carlos support our work all the time. Recently, we initiated a collaboration with Fabienne, which we hope will make our exploration of Baronia even more exciting than our previous work. Year after year, the citizens of a small town generously offer us their houses, their hospitality, hosting us in their homes. These people even allow us to use or transform their homes into our laboratory. We alert their children about the importance of caring for Baronia. My own child enthusiastically assists us in the field. Here he is observing an orange female morph. Mr. Goyo is an exceptionally enthusiastic and skilled collector. He helps us to find new populations every year. Meanwhile, Miss Maria cooks delicious Mexican dishes for us. The town has become actively involved in our efforts, understanding that Baronia is not a threat to them or to their cultivates as they previously thought, but rather something to be protected. Thank you very much for your attention.